On behalf of GlycoNet, welcome everyone to today's webinar about glycan synthesis and analysis. This webinar is part of our current series on GlycoNet integrated services. So each webinar covers a different area of services. If you've missed our first webinar that gave an overview of the facilities and different services available, you can find the link on the webinar section of our website under news and events. You can also follow our newsletter on social media to learn about future webinars and events. If you have feedback or suggestions for future webinars, we just ask that you please fill out our short survey after the webinar. And without further ado, I will now pass off the webinar to our first speaker, Dr. Sicheng Lin from the University of Alberta.
Okay, first, can everybody hear me? Yes? Okay, first I would like to thank our organizing team for putting together this webinar. It's great to be here with all of you today. So my name is Sicheng. I finished my PhD degree in 2020 under the supervision of Tal Ari, where my research mainly focused on the synthesis of complex carbohydrates. And seeking to broaden my experience and skills, I transitioned into industrial and spent around 2 years as a research scientist at a CRO company. And now I'm the manager of carbohydrate synthesis facility at GlycoNet Integrated Services. So in today's session, I will discuss the capabilities of our facility and how it can support and enhance your research efforts. So to give you a bit of background, the GlycoNet Integrated Services was launched by GlycoNet in 2023 as a groundbreaking initiative to advance glycomic research in Canada. We have a distributed service facility across 7 Canadian institutions. This innovative structure allows us to offer a comprehensive suite of glycomic services from glycan synthesis and analysis to structural characterization and function studies. Our mission is to provide highly specialized expertise and state-of-the-art tools to bridge the gap between research and application. GIS aims to accelerate innovation in glycomics and contribute to advancements in areas such as vaccine development, diagnostics, and biotechnology. As a synthetic chemistry facility, our primary goal is to serve as a catalyst for research by providing the essential tools, expertise, and resources you need to accelerate your work. Throughout this session, I will talk about the range of services we offer, as well as share some examples to demonstrate how we have helped clients to overcome the challenges and achieve their goals. To start with, why are we interested in glycans and why are they important? Glycans are the most abundant biological molecules on Earth and are widely present in all living organisms. They play key roles in many biological processes, so understanding glycans is essential for advancing fields like immunology, cancer research, and infectious disease. However, accessing glycans for research is challenging. There are mainly 2 ways we can either extract or purify from natural source or make synthetic glycans. Glycans produced in biological system are heterogeneous. As a result, the natural glycans usually bear the structural complexity and heterogeneity. This means you will end up with a mixture of different glycan structures, making it difficult to study their specific functions. Additionally, the extraction process can be time-consuming, expensive, and usually yielded limited quantities. In contrast, synthetic glycans are homogeneous and structurally well-defined. Synthesis can give a single pure glycan structure, which is ideal for studying its specific biological function. Moreover, synthetic glycans can be produced at a relatively low cost and in large quantity, making them more accessible. This can help people to elucidate the function of glycans and glycoconjugates more effectively. So our synthetic facilities specialize in design, modification, and synthesis of glycans, utilizing both chemical and chemoenzymatic strategies to produce high-quality synthetic glycans for use as biological probes. These probes are essential for understanding the structure, function, and interactions of glycans in various biological systems. In addition to synthesis, we also offer expertise in a customized probe molecule design, Our team can work with you to design and develop the ideal probes for your specific research needs. Moreover, we also are equipped with an automated glycan synthesizer. This state-of-the-art technology allows us to streamline the synthetic process and improve project turnover times. It also allows us to create libraries of related but distinct glycans quickly. Furthermore, we provide contract research services, allowing you to leverage our specialized knowledge and resources to advance your project without the need for in-house expertise or infrastructure. This includes, but are not limited to, research and development on new synthetic compounds, optimization of reaction schemes or conditions, development of manufacturing process, as well as data interpretation. Whether you need custom glycans, assistance with probe design, or collaborative research support, our facility is here to help you achieve your scientific goals more efficiently and effectively. So our process begins with a collaborative dialogue to fully understand your requirements. Because in glycan synthesis, determination of the target molecule is a critical first step, as the structure of the target glycan usually directly impact the complexity, difficulty, as well as the cost of the synthesis process. Once the structure is defined, we will develop a tailored strategy to synthesize the glycans. There are 4 primary approaches we use to make glycans, such as enzymatic synthesis, which is ideal to achieve perfect radio and stereochemical control with unprotected sugars as substrates, and chemoenzymatic synthesis, which can combine the flexibility of chemical synthesis with the precision of enzymatic reactions. And it often simplifies the process and reduces the need for protecting group manipulations. And chemical synthesis can provide you flexibility for creating diverse unnatural structures. However, it will become more labor intensive for the synthesis of complex glycans. And moreover, the automated synthesis can produce diverse glycans with high reproducibility, and is particularly useful for small scale and making linear polysaccharides. So each method has its unique advantages, and the choice of approach depends on the specific requirements of the project, such as the complexities of the target glycans, the desired scale, and the intended applications. So as a result, we carefully consider which synthesis method will be the most cost-effective and time-efficient for a project. So nature has addressed the challenges of glycan synthesis by evolving enzymes, and these enzymes can be used by chemists to catalyze the formation of glycosidic bond in a synthetic context. So implementing enzymes in glycan production offers some advantages, but also bears some limitations. Enzymes can construct glycosidic linkages in high yield with perfect stereo and radio selectivity. The reactions are also more efficient without the need for installing or removal of numerous protecting groups, which are often required in traditional chemical synthesis. Moreover, enzymes typically operate under mild conditions, which minimize the risk of decomposition of sensitive molecules. In addition, enzymatic reactions are generally more sustainable, as they don't need to use toxic chemicals and solvents. However, enzymes are typically less flexible because they're substrate specific and usually less readily available in comparison to chemical reagents. In addition, the nucleotide sugar donors are sometimes unstable and expensive. So in our facility, with the expertise of our scientific director, Warren Walker-Chung, we have built a library of enzymes that are available to use in-house. We have different glycotransferases available. For instance, sialic acid is a critical component of many biological active glycans, playing key roles in cell signaling, immune response, and pathogen interactions. We often work with clients who need to install sialic acid on their glycans, and in those cases, clients will send us their glycan intermediates, and we perform the sialation reactions using our specialized sialotransferase. Once the reaction is complete, we will return the final silated product to the client, which is ready for use in their research. So this collaborative approach allows you to focus on your core work while leveraging our expertise and resources to achieve your synthetic goals. Many successful applications of enzymes in glycan production have utilized chemoenzymatic synthesis, relying on a combination of chemical and enzymatic steps that typically begins with chemical synthesis and ends with enzymatic extension. This is one of the key strengths of our facility, allowing us to produce complex glycans efficiently. For example, making those gangliosides chemically would be quite difficult. However, this lactose core can be achieved easily by chemical synthesis. Then we just need to use 2 or 3 enzymatic reactions so that we can produce those sialic acid-containing glycans in a couple of weeks. More importantly, I want to emphasize that we can handle those enzyme reactions up to gram scale, which means we can meet the demands of both small-scale reactions and larger-scale applications. And here's another example to show how we incorporate enzymes to prepare diverse glycans. So our carbohydrate synthesis platform played a crucial role in advancing a bead-based ABO antibody assay, which is a critical tool for improving donor-recipient compatibility assessments in organ transplantation. This project required the synthesis of a complete set of 18 ABO antigen subtypes. So the antigens expressed on the red blood cell determine an individual's blood group, and ABO antigens are classified with the presence or absence of a terminal sugar residue as shown in blue color. For example, people with O blood type are only able to produce H antigen, whereas individuals who are blood group A or B will have extra sugar residue attached on the galactose. And it can be further subtyped according to the sugar residue, which is present at the reducing end, as shown in red, from type 1 to type 6. So initially, the chemical synthesis was developed by a PhD student 15 years ago. So they required 10 different building blocks in total to make all 18 compounds, which also involved more than 100 steps, along with a lot of protein group manipulations, as well as column purifications. Later, researchers need more materials for their application need, so our synthetic facility stepped in to provide expertise and infrastructure necessary to reproduce those glycans. While remaking those glycans, we also try to streamline the process by installing the terminal sugar with either a GAL transferase or a GlycoNet transferase. It helped us to avoid the preparation of 2 building blocks. And now we are currently trying to incorporate 2 or 3 enzymatic reactions to make them less labor intensive. And in those cases, we only need to chemically synthesize the monosaccharide core for each subtype. Next, I will show you another project that we have been working on, which involves the development of an animal vaccine against Streptococcus suis. Streptococcus suis is a peanut-shaped gram-positive bacterial swarm pathogens leading to important economic losses to the industry worldwide. S. suis is also an emerging zoonotic agent, meaning it can infect humans. In recent years, the number of reported human S. suis cases has substantially increased. The control of this disease is mainly attributed to the overuse of antibiotics, which actually leads to a dramatic increase of the risk related to antimicrobial resistance. From a One Health perspective, vaccines should be a key component of strategies for preventing S. suis disease and reduce the threat of antimicrobial resistance. So however, there are currently no commercial vaccine available. And S. suis is an encapsulated bacteria covered by a capsular polysaccharide. The CPS is proven to be the essential virulence factor that protects the bacterium from the host immune response. The repeating unit structure of the CPS has been resolved, and the researchers proposed to develop a synthetic glycoconjugate vaccine, which consists of 2 components, the carbohydrate antigen and carrier protein. The carrier protein is used to overcome the poor immunogenicity of pure carbohydrates. The preparation of this glycoconjugate started from the synthesis of this heptasaccharide. The initial development involved a collaborative efforts by 4 postdocs in a research lab. However, while they moved on to the next stage of their careers, reproducing those glycans become a major issue, hindering further immunological analysis and vaccine development. Then our synthetic facility took over this project, reinvestigated the synthesis, and helped our client to reproduce more material to continue their work. The synthesis of this glycan contains more than 40 chemical steps and 2 enzymatic steps, along with over 30 column purifications. And now we are involved in developing the potential pipeline to manufacture the vaccine. And to this arm, we will optimize the entire synthetic strategies to reduce steps, simplify the purifications, and increase the overall yields to make it more cost-effective and scalable. So I believe those two projects are perfect examples of how GIS bridge the gap between research and application. They showcase our capabilities to solve complex synthetic challenges and translate scientific discoveries into practical applications that benefit society. Lastly, our facility also equipped an automated glycan synthesizer, which is a powerful tool that expands our capabilities in glycan production. It is a solid phase synthesis, so there are different resins available, such as producing a glycan with a free reducing end, or with an alkyl spacer, or with an amine spacer. What I've shown here is a photocleavable linker that allows you to convert the glycan to glycopeptide, glycoconjugate, fluorescent probes, or immobilized on microarrays for the high throughput analysis of protein carbohydrate interactions. So just want to give you some ideas in terms of scale and type of molecules we can make by this instrument. The default resin scale for this instrument is around 40 milligram. So after some tests, we figure out that the maximum loading is actually around 80. So what does it mean? It means you will probably get 1 to 5 milligram of final deprotected product by the end. So for now, the major limitation is actually the availabilities of those building blocks, which mainly sold by GlycoUniverse. And the cost of them are also not cheap. But overall, it's good to produce linear oligo and polysaccharides, such as alpha-2,6 polymonosaccharides. And in addition, I think it is best to produce libraries of related but distinct glycans in small quantities. Here, I will just briefly describe how this instrument works, starting from the bottom part of the instrument. So on the left side is where all the solvent bottles will be placed, and the right side will be all the chemical reagent bottles. The top middle part is the reaction vessel where all the reactions will take place. And to the left of this reaction vessel, it's a rotating disc where you can put all your building blocks. And to the right of the reaction vessel, it's a fractional collector that mainly are used to either recover your building blocks after glycylation or collect your compounds after a photo cleavage. So this is a video to show you how it works. So this yellow particles are the resin. So the solvents, reagents, and building blocks will be dispensed into the reaction vessel, and the reaction will be mixed by argon. And after each step is done, the side products, reagents, and unreacted materials will be washed away, while the desired glycan will remain covalently attached to this resin. So once all the synthesis steps are completed, the glycan will still attach to the solid support via a photo-cleavable linker. So to release this glycan into the solution, we use 2 UV lamps with wavelengths of 370 nanometer. These lamps are positioned to shine directly onto the reaction vessel. And you can also decide how many cycles you would like to do for the cleavage. And after each cycle is done, the solvent that contains your product will be transferred into a test tube in the fractional collector, which are ready for purification. So a standard workflow will be, if you want to make a tri or tetrasaccharide, for example, you just need to set up the instrument And the next day, the reaction will be all done, and you can do the photo cleavage and purify your compound by HPLC. To conclude my talk here, I want to emphasize that our team is fully committed to leveraging our expertise and state-of-the-art tools and innovative approaches to support your research. Whether you're exploring fundamental glycobiology or developing glycan-based applications, we are here to help you achieve your scientific goals successfully. So if you have any questions or concerns or would like to discuss how we could assist you with your project, please do not hesitate to reach out to us at this email. And we look forward to working with you and contributing to your success. And thank you for your attention. So, any questions?
If you think of any questions, feel free to put them into the chat. Otherwise, if there are no immediate questions, we can move on to the next presentation by Dr. François Le Mauff.
Yeah, I will close my...
You're on mute.
Of course, this still happens now. So, as I was saying, so my name is François Le Mauff. I'm based in Montreal, Quebec, and I'm the node manager of a glycan analysis node, but I'm focusing mainly on a specific type of glycans, the one produced by microorganisms. So here is what we can do for you. So as I said, and this is supposed to start now. So my node is specific to microbial glycans, so everything that is made by little bugs that wants to kill us. And as those pictures are not really representing like what I'm doing, I ask our favorite new toy of AI to say like, okay, what can you show me when I say like microbial glycomics? It didn't start to give me anything, so I tweaked a bit the keywords with fungi, bacteria, sweets, and this is what it came up with. So most of those pictures are not really accurate, like or fungi and bacteria do not look like those candies. But I have to say that this last shot right there was actually not too far from the reality, and why should we care about those microbial glycans? Because if we take those little like cupcake fungi covered with icing sugar, well, if we slice through them as if we slice through our bacterias, mycobacterias, or fungi of interest, then this is what you'll see. Like all those organisms are actually covered and coated and encapsulated in a thick layer of sugar, the same way those cupcakes are coating with this icing.
Sorry,
And
François, could you just speak a little louder or closer to your mic? Just
okay,
some noted
it's
that there.
okay, sorry for that.
It's a little bit quiet. Thank you.
Okay, is it better if I'm right here? Okay. So I was saying that those microorganisms are actually completely covered and coated with a thick layer of sugar. And that when we start to dig through those different bacteria or fungi here, for the example, we are facing wide diversities of polysaccharides that are out there. And the same way that when you eat those little cupcakes, like the first thing that you're going to taste is this sugar coating. Well, the first thing your immune system is actually tasting when you are having an infection is this sugar coating of all those bacteria or different organisms that wants to kill us. So this is why it is important to look at microbial glycan. It's quite different from regular N and O glycans that we just heard about, but this brings a whole lot of opportunities and a lot of expertise to go after those sugars. So what we can do for you if you're interested in microbial glycans, that we can look at basically almost everything. And I like to think that the way my platform operates is not just like we're having a request, like we need to titrate this or we need to make this and I'll do it and I'll say bye. I like to get involved in the project and collaborate as much as I can to help you as much as I can with this expertise in terms of microbial glycomics. And I can look at stuff like composition and structures of sugars, like we have all the MS and LCs technology to do this kind of thing where we can break down sugars and reconstitute the structure of those different glycans from bacteria or fungi or parasites or extracellular vesicles, so on and so forth. I can also look at all different enzymes that are involved in synthesis, modification, or degradation of those polysaccharides. And here's an example we just published with a group at SickKids in Toronto, Ontario, where we looked at the biofilm, at a polysaccharide critical for biofilm formation in Pseudomonas aeruginosa. And after purification of the substrate, I was able to use those different enzymes to reconstitute the synthetic pathway of the polysaccharide within the periplasm. Or I can also look at interactions of those polysaccharides with different proteins. And here's an example of a project that is currently ongoing, where we're looking at new molecules called lectibodies, so a part of lectin and a part of antibody that could be of use in our fight against here Cryptococcus neoformans. And as you can see, those lectibodies are pretty good at recognizing them. So we hope that they're gonna be able to also be really good at getting rid of the yeast once they are in our systems. So instead of me continuing to speak about what I can do for you and how I can help, I was thinking that if I ask two trainees that actually use the platforms to describe what they are doing, how the platform was important, it will be way louder than me going through the different things that I can do for you. So I have the great pleasure to have two trainees to join us here. CM from McGill University working on TB, and Nicole from Dartmouth University down south in the US that works on Aspergillus fumigatus. And as you'll see, it's two different projects, two different polysaccharides, two completely different stories, but the platform was helping them with their stories. So without further ado, ladies, I'll let you take it away. And maybe CM, you can start as your pictures ended up like first on the slide.
Yes. Yep. Let me share. Okay, perfect. Is it on?
Yes.
Okay, hello everyone. Today I'm gonna talk about the characterization of the mycobacterial peptidoglycan using MALDI-TOF. So to start with, I'm going to do a small introduction about TB. So also known as the consumption during the 19th century, it's an infectious disease that's caused by the bacteria Mycobacterium tuberculosis, characterized and identified by Koch by the end of the 19th century. He did not find any cure against the disease until Smith characterized bovis. So Mycobacterium bovis is the bovine form of TB. This bacteria was really helpful since it gave us the first vaccine against TB. So TB is still the deadliest infectious disease from the last years, with COVID being in front in the last few years, but it came back first. I don't know if it's a good thing, but it came back first again this year. And the only licensed vaccine against TB is still BCG, which is the vaccine that is a live attenuated form of Mycobacterium bovis. So till this day, 100 million doses are given each year to neonates and infants, but its protection is really uncertain, going from 0 to 80%. And one of the reasons why it is so low is because the mechanisms underlying the protection against TB are still unknown. And one of the things that we focus on in my lab is the peptidoglycan. So one of the specificity of mycobacteria is its cell wall. So mycobacterial cell wall is really thick. It's composed of multiple complex layers that are really rich in lipids, giving it a lot of resistance against antibiotic. Therefore, we have really long treatments against TB. And against other bacteria, mycobacteria differ specifically in its peptidoglycan. So the mycobacterial peptidoglycan is composed of repetition of glucanac and glycolyl muramyl dipeptide. That I don't think I have my laser on. Okay. So this is very particular because most bacteria have a peptidoglycan composed of a repetition of glucanac and N-acetyl muramyl but mycobacteria has a glycolylated peptidoglycan. And the only difference between these two is due to a gene called NAMH that encodes for an hydroxylase that adds an hydroxyl group to that MDP. So the only difference between the N-acetyl MDP and the glycolyl MDP is that hydroxyl group caused by NAMH. And what is really interesting is that NOD2, the intracellular receptor of TB, recognize that N-acetyl MDP. But in the literature, it has been shown that the N-glycolyl MDP has an increased recognition by that receptor. So that's why we focused on the peptidoglycan to better understand the interaction between the host and the pathogen. So first off, we studied Mycobacterium smegmatis, which is a strain of mycobacteria that is way easier to work on, basically. It grows faster and it is easier to do knockouts, knock-ins in it, etc. So previously in the literature, Raymond in 2005 that identified the NAMH gene that causes that modification of the peptidoglycan showed with GCMS that in his wild-type strain of smegmatis, he had a mix of acetylation and glycolylation with a higher ratio for the glycolylation from 7 to 3 for the acetylation, while in his knockout of NAMH in Mycobacterium smegmatis, he had a complete acetylation. In our hands, we did a chemoenzymatic preparation with François. We extracted and purified the peptidoglycan. We then did a metanolysin digestion, and then we shielded through MALDI-TOF the samples. And what we found is that in the wild-type strain, we had a complete glycolylation of the peptidoglycan. So we were doing, okay, that makes sense. And for the knockout, we had a complete acetylation. So that made sense too. And what's really cool is that we could superpose those two spectrum. And the only difference between these two is the mass of that hydroxyl group that's added by NAMH. So that was cool for Mycobacterium smegmatis, but it is not a member of the MTBCs, or the Mycobacterium tuberculosis complex, so bacteria that causes TB. So we went into members of the MTBC. In the literature, what we had is for Mycobacterium tuberculosis, previously in our lab, an RPH PLC has been done with Mycobacterium tuberculosis and Mycobacterium tuberculosis delta NAMH. And what they found is very similar to what we had in Mycobacterium smegmatis in the literature. So a mix in the wild type at the top that you can see here of acetylation and glycolylation. While in the delta NAMH strain, we had a complete acetylation of the peptidoglycan. And in our hands, we had BCG, so the vaccine against TB, that's a level 2 bacteria, so again, easier to work with, but also very similar genomically to Mycobacterium tuberculosis. And in our hands, Mycobacterium bovis, BCG, showed a complete glycolylation in the wild-type strain, while we had a complete acetylation in the knockout of NAMH. So again, same thing. The only difference between these two spectrum is the mass of that hydroxyl group. So we confirmed with François an efficient protocol to characterize mycobacterial peptidoglycan. So confirmed in non-tuberculosis mycobacteria with Mycobacterium smegmatis. We also did it in Mycobacterium abscessus, another strain. We confirmed it in a member of the MTBCs, so Mycobacterium bovis BCG. And therefore, the experiments following were associated with the glycolyl MDP of our peptidoglycan, since we could see differences ex vivo with, for example, macrophages experiment with a differential immunogenic response when we infect with a knockout of NAMH or a wild-type strain. In vivo, we also did experiments of vaccination with our BCG strain, for example. We couldn't see any difference in terms of protection, so that gives us also a lead on the role of the peptidoglycan and NAMH in that aspect. And in the future, that protocol is going to be extremely useful to characterize other members of the MTBCs, such as Mycobacterium tuberculosis that we have in the lab, but that's in the CL3, in a level 3. So we need to confirm the protocol there too. We can do that also with Mycobacterium bovis, with Mycobacterium oryges, zoonotic strains of TB, but also in other environmental bacteria such as Mycobacterium kansasi, Avium hominisuis. So a lot of strains. So we have a really good protocol to study a lot of various aspects of mycobacteria, such as virulence, vaccination, and drug treatments. So thank you, everyone. If you have any question, I'll be very happy to answer them. Cannot hear you, François.
Thank you, CM. I guess we can get the question at the end. So we can just use the flow of the presentations. So after showing this demonstration of a platform in extracting, purifying, and analyzing like mycobacterial and bacterial polymer, let's just switch gear and go into the fungal kingdom with Nicole. Nicole?
Hello, everyone. Thank you so much to François and GlycoNet for the opportunity to talk about what I've had the opportunity to do with François. So today I'm going to be talking to you about the fungus Aspergillus fumigatus and how the extracellular matrix can vary based on which strain you're looking at and how that has implications for disease. So Aspergillus fumigatus, just a quick little introduction. It's a filamentous mold, and it largely reproduces asexually. So I would want you to hone in here are just the little dots that are present at the top of the blue structure there. They are present ubiquitously. If you walked outside today, you most likely breathed some of these spores in. However, this is not really a problem for people with an intact immune system. They can easily be dispersed within the air, and when they're inhaled, your innate immune system is able to clear these canidia and you avoid disease. However, this is not the case for immunocompromised patients, where this can lead to various forms of pulmonary mycoses and has a high mortality up to 90%. So interestingly, when these canidia are inhaled, they are able to swell in a mode of growth called isotropic growth, and they eventually form these germlings that produce biofilms. And as they grow, they are consuming oxygen and generating these hypoxic environments. And this is a really big interest for our lab, as we've previously shown that strains that grow better in these low oxygen conditions are more virulent in a murine model of pulmonary aspergillosis. So what's really beautiful is our fungus here grown on a plate AF293 is our wild type, showed this beautiful flat velvety colony. However, strains that are hypoxia fit take on these really furrowed kind of ugly morphologies. But what's really interesting about this is these can be isolated from diverse patient populations, including patients with invasive pulmonary aspergillosis, patients with cystic fibrosis, or even patients with B-cell malignancies or other forms of cancer. So we've termed this really kind of crazy, cranky morphology H-morph for hypoxia-related morphology. And we've defined a set of criteria that's important for these, including displaying hypoxia fitness, having more than 3 of these furrows, which are kind of its wrinkles radiating from the center, and greater than 40% vegetative mycelia, which is that white rim, as opposed to the green area, which harbors the little circular conidial propagules. So we used a directed evolution approach to identify a single nucleotide polymorphism in this uncharacterized gene that's sufficient to cause this weird morphology, increase hypoxia fitness, and most importantly, it causes increased virulence in our murine model. But we really aim to figure out why this is, and that's where my whole project sort of focuses. So one of the phenotypes that we were really interested in the beginning is this hypoxia fit strain HRMA-Rev is significantly less adherent in vitro. And this was kind of confusing because when we think about microbes, one of the number one virulence attributes is the ability to adhere to promote disease. And that's the opposite of what we see here, despite it being more virulent. So adherence for Aspergillus fumigatus is largely mediated by the extracellular polymatrix, which is largely composed of the polysaccharide galactosaminogalactan. So François did a much better job than I am, but here is my beautiful little cartoon of the green depicted GAG or extracellular matrix that's covering all of the other polysaccharide polymers at the cell wall, which are highly antigenic. And we can see that this GAG polymer is produced in vivo, which is really important. And more importantly, when you delete the ability of a strain to produce GAG through the deletion of UG3, there's a significant reduction in virulence. So going back to my H-morph strain, what we had previously noticed is that this strain is actually capable of producing an extracellular matrix, but it doesn't really well adhere to the fungal cell wall. And we're not entirely sure at the moment why that is, but it's something that we're actively looking into. So just to go back a quick step, I just wanted to talk about some of the key players in the ability to produce this extracellular matrix. And I'd love to focus on the little green circle down at the bottom, which is an enzyme required to convert our sugars to be processed to then be spit out into the extracellular matrix. And at the moment, it's thought to be bifunctional. But that's up for debate. But more importantly, when you delete this gene, you completely ablate the ability to produce your extracellular matrix. So we deleted this gene from our really wonky H-morph, and we decided to put it back into our murine model. So you'll see for our PBS, we had no mortality. For our N-morph or wild type AF293, we saw what was expected. When we deleted the ability of that strain to make GAG in our murine model, we didn't see much of a significant difference. However, I would like to highlight that our model differs from the one that I had previously showed you, which could contribute to this. For our H-morph, we saw a significant increase in virulence, but when we deleted the ability of this strain to produce the extracellular matrix, we saw that its virulence profile looked a lot more like the flat colonies. And when we reconstituted our UG3 back into our H-morph, we saw the restoration of virulence. So this led us to hypothesize that there's something different about the composition of the extracellular matrix that's produced by our H-morph strain that results in this increased virulence. So to look at that, we turned to our wonderful collaborator, François, where we were able to produce fungal cultures in shake flask cultures and then isolate and purify out the extracellular polysaccharide matrix, which we then treated to look at either soluble or insoluble fractions and performed microbial glycomic analysis via GCMS. And while this is a beautifully summarized tiny slide, I want to highlight that this takes weeks and it was no small feat on François' part and really highlights how heterogeneous not only the polymers can be, but also this organism, which you will see by the results on the next slide. So here we were able to quantify different polymers that were found in the extracellular polysaccharide matrix. And what I really want to highlight here is actually in this teal color at the man and the gal portions, which likely denotes an increase in galactomannan content, which is a highly antigenic sugar for Aspergillus fumigatus. So currently we are in the process of getting some repeats so that we can hopefully bring down some of this variation, which is largely just due to the variation in the growth of the fungus under these conditions. It can be rather finicky, in hopes that we can identify polymers that might be driving increased virulence. So we can then go back to our murine model and manipulate that to see how we might be able to develop further treatments for this fungus, especially as it relates to this morphotype. So with that, I'd love to thank my lab and really François for all of the incredible help in helping me understand polysaccharides and glycans in general. None of this would have been possible without him and the support of GlycoNet. So thank you, and I'm happy to take any questions.
Thank you, Nico. So this is an overview demonstration of the heterogeneity that microbial glycan can bring and what I can do for you or what I can help you realize. And I also wanted to give a shout out to new services that my platform will be offering. So something completely different from microbial glycans, but critically important too in response to infectious disease. It's the glycoproteomic services, something that is new and currently under development under my node, but already accepting projects. So if you have any kind of proteins of interest that you would like to know what kind of N glycans or O glycans or at the surface and what's the site occupancy or any kind of over question, just don't hesitate to contact us. So either through the GIS website or through this QR code out there, it's gonna give you directly my contact if you scan it. And I'll be able to help you and really happy to help you with any kind of microbial polymer that you can have. Just don't hesitate to throw it at me. I've started like brand new those protocols on mycobacteriums and got this great results for CM and ER projects. Aspergillus was where I started off, but all projects are bringing something new, and I'm not scared of facing any kind of new parasites or new bugs and look at those sugars. So with that, I'll take any questions for any of us, I think. Thank you.
While we give people maybe a moment to write in chat if they have any questions, I just want to thank everyone for joining us today. A link to the recording will also be shared via email, and we'll be organizing more webinars on different areas of our services. So make sure to sign up to our newsletter, follow us on social so you can find out when the next one is scheduled. And we really appreciate it if you take the time to fill out the short survey from today as well. If there are no questions, I think we'll conclude today's session. Thank you so much, everyone, and thank you to our presenters today as well.
Thank you, everyone.
Thank you.
Thank you. Thank you.